Okay, this is a continuation of the lecture that I started earlier today. Specifically here, I'm going to cover pages 260 and 261. So we're going to talk about the three alternative models for DNA replication, and then we're going to talk about the Meselson and Stahl experiment. So in April of 1953, when Watson and Crick published their paper about the structure of DNA, the double helix, they proposed in that same paper that the copying mechanism for DNA is semi-conservative replication. And all that means is that the two strands of DNA must separate before they can be copied, and that each of those two strands then serves as a template or a pattern that determines what the bases are going to be on the new strands of DNA. And that'll make more sense. If you don't remember how that works, that'll make more sense when we look at the pictures. But keep in mind, the semi-conservative model is the one that was preferred by Watson and Crick. It's the one that they thought was correct. The problem is they could not prove it. There was no way to prove that that is how DNA is really copied, and there were two other models, the conservative model and the dispersive model, which had been proposed by other people, and nobody really had a way to definitively prove which model was correct and which of the models was incorrect. Until 1957, when Matthew Meselson and Franklin Stahl came up with a way once and for all to prove two of these models to be incorrect. And they were able to prove that one of the three models is in fact the correct model. So we need to try to understand what these three models are, what they say about how DNA is copied. So first you have the conservative model. And remember, in all three of these models, in all three pictures, the dark blue color represents the old original strands of DNA. So we're looking right here at a double helix. Here are the two original strands of DNA that we're trying to copy. And after we copy it, the light blue represents the new DNA, the brand new copy that was just made. And so the conservative model says that when you copy a double-stranded DNA molecule, that the two strands never separate. They remain connected. So it's almost like a copy machine. If I was to put this double helix into a copy machine and hit the green button, it's going to spit out an exact copy. It'll be a new copy, but it'll be exactly like the original, but I'll still have my original, right? So if you put a piece of paper on a copy machine and you make a copy, yes, you'll have the new copy, but you'll still have your original. So that's how the conservative model says DNA is copied. And it's kind of hard to imagine how that would really work in a cell, but it was a proposed hypothesis. So now that I've made a copy, if I was to copy the DNA again, I would get another new copy plus the copy that I just made a minute ago, and then I've still got my original, and then the copy that I just made of the original. So if you take one DNA molecule and you copy it once and then you copy it again, what are you going to have? You're going to have the same original double-stranded DNA that you started with, and you'll end up with three newer copies. Okay, that's the conservative model. The semi-conservative model, remember, this is the one that Watson and Crick were in favor of. They couldn't prove it, but this is what they thought really happened, was that the two original strands of the DNA first have to separate. So you see there's a dark blue strand here and a dark blue strand here. Those are the two original strands of the molecule that we're copying. They said, okay, so those two strands separate, and then a brand new strand is built for each of the DNA molecules. So when you're finished, you have two DNA molecules. They're exactly the same. They're identical to each other, but each one has an old strand and a new strand, an old dark blue strand and a new light blue strand. And if I was to copy both of these again, I'm going to separate the two strands. So the dark blue goes here, the light blue goes here, and then I'm going to fill in with another new strand, another new strand. And I'll do the same thing here. So based on the semi-conservative model, if you copy the DNA once and then twice, you'll get 4 DNA molecules. 2 of them will look like this. They'll be half old, half new, and then 2 of them will be completely new. And then the strangest model of all is called the dispersive model, which basically says in order to copy this double-stranded DNA, you first just have to chop it up into little pieces. So notice you've got these little fragments of dark blue that are just kind of scattered evenly around, and then the gaps are filled in with brand new DNA. It's the dispersive model because you're basically chopping up the DNA and dispersing the pieces. But every time you copy the DNA again, you're going to get more and more of the new DNA and less of the old. So if you look here, all 4 of these have little pieces of old DNA, but they're mostly light blue. So those were 3 different models, 3 different hypotheses about how DNA might be copied. And the problem is you cannot, well, even today with modern microscopes, you can't just look into the cell and see how the DNA is being copied, because old DNA is not really dark blue and new DNA is not really light blue. They are exactly the same color. And so it's hard to tell what's really happening in the cell. So this was a real problem. How are we going to figure this out? How can we tell the difference between old DNA and new DNA? So the 2 men who figured it out, Matthew Meselson and Franklin Stahl, came up with an ingenious way to tell the difference between old DNA and new DNA. And it wasn't by color, it was by mass. So they figured out a way to make the old DNA heavier and the new DNA, what we see here as light blue, as having less mass. Now, how did they do that? Well, we'll get to that in a minute. Let's take a closer look at the 3 models that we just went over. So I'm going to show you the same picture again, but this time I've annotated it a little bit. Hopefully this will make it a little more clear what we're saying. So the conservative model, if it's correct, says that once you copy it, once you copy the DNA, you're going to have the old original double-stranded DNA that's still completely old DNA or original DNA, and then you're going to have this brand new copy that's 100% completely new DNA. So if I look at all of the DNA now, exactly half of it is old and exactly half of it is new. And then if I copy again, I've still got my original. So 1/4 of my DNA at this point is that old original double-stranded molecule. But I've got 3/4 or 3 newer double-stranded DNAs. Okay. The semi-conservative model, the one that says, okay, we're going to take these 2 old strands and separate them, and then we're going to fill in with new DNA, it says after I copy the DNA once, all of the DNA, both of these DNA molecules, are going to be an exactly 50-50 mix of old and new. 1 old strand, 1 new strand. 1 old strand, 1 new strand. So the DNA, every bit of the DNA should be a 50-50 mix of old and new. Of course, the dispersive model also says that after copying one time, all of the DNA should be a 50-50 mix of old and new. It's just arranged differently. Instead of one old strand and one new strand, it's little pieces of old mixed with little pieces of new. But then you can see the difference when you get down here. After the DNA is copied twice, you see a real difference between the semi-conservative model and the dispersive model. Because the semi-conservative says if we look at all four of these DNA molecules now, half of them, 2 of the 4, should be a 50-50 mix of old and new, but 2 of the 4 should be 100% completely new, shown by the light blue. And the dispersive model says after copying twice, all of the DNA, all 4 of them, should be mostly new DNA with just little pieces of the original dark blue remaining. Well, remember, DNA is not really color-coded. So Matthew Meselson and Franklin Stahl had to come up with a really ingenious, creative way to tell the difference between old DNA and new DNA. So let's look at the Meselson and Stahl experiment from 1957. This is, your textbook has a really nice illustration of this that's perfectly good, but I thought this might be an easier way for you to understand exactly what these two guys did. So they worked with E. coli bacteria. And E. coli, or any bacteria really, is a good choice for this because bacteria reproduces very quickly. Every 20 minutes, a bacterial cell copies all of its DNA and then divides. So every 20 minutes, a copy of the DNA is being made. That's a very fast copying process. And they chose E. coli specifically because it's very easy to grow large amounts of E. coli in the lab. So what they did is they grew E. coli bacteria for several generations, like maybe several days. They grew the E. coli in petri dishes that contained nutrient. It's called nutrient agar. It's the food that the bacteria are living off of. But this food was special. It contained nitrogen 15, which is a heavy isotope of nitrogen. Normally, nitrogen has 7 protons and 7 neutrons, which would be an atomic mass of 14. So this is heavy nitrogen. And if you feed heavy nitrogen to bacteria, they will build their adenines and guanines and cytosines and thymines out of heavy nitrogen, and so it literally makes the DNA a little bit heavier. It has a slightly greater mass than normal regular DNA. So they grew this bacteria for many generations so that all the bacteria has this heavy DNA. Then they took a portion of these bacteria, some of the E. coli, they took them out of this container with the heavy nitrogen, and they transferred them into a new container which has nutrient medium with regular, normal nitrogen 14. So what does that mean? It means any new DNA that is made in this new container is going to be lighter. It's going to have a lower mass. So again, all the bacteria has heavy DNA, but if you take just a few of those bacteria and you transfer them into this new container, any new DNA that they make is going to be light. It won't be a different color, but it'll be a different mass. So the first time they did this, they only left the bacteria in the light nitrogen for 20 minutes. The first time they did it, they only left them in there for 20 minutes. So that's exactly enough time for E. coli to make one copy of the DNA. 20 minutes allows the E. coli to copy their DNA one time. Okay, the second time they did this, let me get rid of these test tubes because I don't want to confuse you yet. The second time they did this experiment, when they transferred the bacteria into the light nitrogen, they left them in there for 40 minutes, which is enough time to copy the DNA twice. Okay. Meanwhile, they took some E. coli out of the heavy nitrogen and they extracted the DNA from those bacteria and they put it in a test tube. Here's the test tube. They also put a liquid, it's a type of salt solution called cesium chloride. It's a really thick salt solution that they mixed in here with the DNA. And then they put the test tube in a centrifuge to make it spin really, 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 really fast. And the heavy DNA, it was slightly more dense than the cesium chloride, and so that heavy DNA moves down to the bottom and forms a band. Because it's so heavy and dense, it sinks to the bottom. Then they took some light DNA out of this container, and they put that in another test tube, and the light DNA is less dense than the cesium chloride, so it floats to the top. So this was just the control. Okay, so they wanted to see if we spin heavy DNA and light DNA in cesium chloride, where will it end up in the test tube? Heavy DNA goes to the bottom, light DNA goes to the top. That makes sense. Okay, now here are your experimental test tubes. The first test tube right here, this has DNA from bacteria that has only been in the light nitrogen for 20 minutes. Okay. They extracted the DNA from these bacteria, put the DNA in the test tube with cesium chloride, put it in the centrifuge, and they wanted to see where is this DNA going to go. Is it going to be heavy DNA? Is it going to be light DNA? And what they got was, drum roll please, they got a band called an intermediate band. Let me label that for you. I'm going to try to anyway. Let's switch to my pen. And I'm going to try to write, this is so hard to do on this computer, INT, intermediate. It's not a heavy band, which would be way down here. It's not a light band, which would be up here. It's right in the middle. So, what does that mean? Well, let's look at the picture again. Let's go back. Bear with me here. If the conservative model was correct, then half of the DNA should be heavy and should go to the bottom of the test tube. And half of the DNA should be light, and it should go to the top. So if the conservative model is correct, instead of getting one band in the middle, they should have gotten a light band, and they should have also gotten a heavy band. But they didn't. So what that meant was they could rule out the conservative model. There's no way the conservative model is correct. But both the semi-conservative and the dispersive model both predict that after the DNA is copied once, all of the DNA should be half heavy, half light. It should be a 50-50 mix of heavy and light. So it wouldn't go to the bottom because it's not completely heavy, and it wouldn't go to the top because it's not completely light. It would go to the middle, which is what it did. So far we've ruled out the conservative model, but we still have these other two models in play. So let's go back. So remember they had bacteria that had been in heavy nitrogen for a long time. So all the original DNA, the old DNA, was heavy. They transferred the bacteria into light nitrogen, and the second time they did this, they left the bacteria in the light nitrogen for 40 minutes, which is enough time to copy the DNA not just once, but twice. They wanted to see, okay, let's see what kind of DNA we get. Is it heavy? Is it light? Where does it go if the DNA has been copied twice? So After 40 minutes in the light nitrogen, they extracted the DNA from the bacteria. They put the DNA in this test tube along with the thick cesium chloride, and they spun the test tube in a centrifuge really fast so that anything heavy goes towards the bottom, anything light goes towards the top. And look at what they got. They got an intermediate band, just like they had gotten over here, and then they got a light band. They did not get a heavy. Okay, now let's think about what that means. If the dispersive model had been the correct model, then where would all of this DNA go in that test tube? Every single bit of the DNA should have been mostly light, with just little bits of heavy. In other words, if the dispersive model had been right, they should have gotten just one band. It would have been an intermediate band. It would have been a little higher. It would have been maybe right along in here someplace, because it would have had more of the light and less of the heavy. But it would just be one band. It would look a lot like this one, except maybe a little higher, a little closer to the light end. But see, that's not what they got. Meselson and Stahl got the result that you see here, which is an intermediate band and a light band. Now, let's go back to the models. I know this is kind of aggravating, flopping around here. The reason they got a light band is because the semi-conservative model is right, and so half of the DNA, 2 out of the 4 DNA molecules, are now completely light. All of this DNA is going to go to the top. But the other 2 DNA molecules, this one and this one, are a mixture, a 50-50 mix of heavy light. So where's that going to go? It's going to go to the middle of the test tube. So half of the DNA is going to go to the middle of the test tube. Half of the DNA is going to go to the top. That's exactly what they got. So this result right here, after 20 minutes, allowed them, Meselson and Stahl, to rule out, to disprove the conservative model. And then, this result, after 40 minutes in the light nitrogen, allowed them to rule out the dispersive model. If the dispersive model had been correct, they would have gotten one band, it would have been an intermediate band, and it would have been slightly skewed towards the light side, if that makes sense. So what does that mean? It means the semi-conservative model is correct. It means that Watson and Crick had been right all along. And there is a wonderful animation that I'm going to try to post on Google Classroom along with this video that explains this a little in more detail. And I just don't know if it's going to work on the Chromebook. I had some problems getting it to work on my laptop, so we'll see. I'm going to try to post it. If it works, great. If not, I think you'll be okay. But also, oh gosh, let's see. Okay, I also wanted to show you the images from your textbook, because now that we've kind of gone through this, these images might make a little more sense to you. So here is the container containing the heavy isotope of nitrogen. This is where the bacteria started out, and then eventually bacteria were transferred over to this other container with light nitrogen. And remember, once they got into the new light nitrogen, they were either left in there for 20 minutes or 40 minutes. The first time they were left for 20 minutes, and then the second time they were left for 40 minutes. So after 20 minutes, when they extracted the DNA from these bacteria, put it in the test tube with the cesium chloride, and they spun it in the centrifuge, they got one band. They did not get a light band, they did not get a heavy band, they got an intermediate band. That result is consistent with both the semi-conservative model and the dispersive model. It is not consistent with the conservative model. The conservative model would have predicted a light band and a heavy band. The second time around, they left the bacteria in here twice as long for 40 minutes. Then they extracted the DNA, spun it in a centrifuge, and they got two bands, a light band and an intermediate band. Now, if the dispersive model had been correct, they would have only gotten one band, and it would have been kind of in the middle, but a little bit more towards the top. It would have looked exactly like this. See where my cursor is there. And that's not what they got. They got an intermediate band, and they got a light band, which means the conservative model is the right model. Watson and Crick, as I said before, were correct all along. The dispersive model is incorrect. The conservative model is incorrect also. So just really quickly, because we're going to go through this in a lot of detail tomorrow, the semi-conservative model simply says that when DNA is copied, the two original strands of DNA are separated from each other, and the A's and the C's and the T's and the G's on those old strands serve as a template or a pattern to direct what the new strands are going to look like. So here's a brand new strand of DNA that's being built to match up with this old strand. Here's a brand new strand that's being built over here. Notice if there's an A on the old, a T on the new. If there's a C on the old, a G on the new. And so it's complementary base pairing. The letters on the old strand determines what the letters are going to be on the new strand. So that's semi-conservative replication. You're going to read more about that tonight, pages 261 to 264. And tomorrow's lecture, which hopefully will not be as long. I am so sorry this took me so long. I'm just struggling today. But tomorrow's lecture should be shorter, and we'll go through the process of replication. Hope you're doing well. See you soon, I hope.